Okay. It's my great pleasure to introduce Ariel Rokem. Um, he is I guess, longstanding, pretty distinguished data science fellow at the UW eScience Institute, um, specializing in statistics and coding and just like general community support and training across UW for data science applications. And he's also a research associate professor in the Department of Psychology. Um, his work involves developing and uh, devising novel kind of analysis methods and packages for neuroscience applications. More specifically, I think you are focusing a lot of your work on fMRI and I guess structural MRI data sets, but he's mentored a lot of neuro folks, including me, for the eScience incubator program. So he's familiar with a lot of the fluorescence recording methods that our center uses. And today he'll be talking about a primer, like introduction on using dimensionality reduction methods on high dimensional data sets, and also getting a little bit, I guess, into some of the more like introduction of the more advanced methods. So with that, Ariel, we can take it away.
Thanks, Charles, and thanks everyone for being here on this late Friday afternoon. Yeah, the presentation today will be about how to simplify high-dimensional data with what I'm going to call linear decomposition methods. It'll become clear soon why these are linear and why they're decomposing the data. I'll start with a little bit of motivation. Neural data is often high dimensional. What does that mean? We're measuring many neurons simultaneously in some of the data sets that I work with. Maybe we work with many subjects where we're sampling from multiple different locations, many different time points, conditions, etc. And that means that the data that we look at is of high dimensionality, and that can complicate things quite a bit. But for many cases, underlying causes of the data are relatively low dimensional. And so if we want to understand a neural system, that often entails understanding the slow dimensional structure, sort of reducing down the dimensionality of the data from the measurement, which can include many pixels, many electrodes, many voxels in the brain, and so on, to network circuits that are maybe just a few in there that operate together within the data that we're looking at. And if that facilitates, of course, understanding, and it can also improve the accuracy of our description of the system, because a lot of what's in the very high dimensional data can be noise that we're trying to kind of smooth away by going to lower dimensionality. Before I go any further, I just want to mention, if anyone has any questions at any point, definitely feel free to just unmute yourself and ask your question. Hopefully there'll be a bit more of a conversation rather than me just speaking to you that way. So if anything comes up at any point and you're like, okay, what does this mean, or what are you talking about? Just please unmute yourself. And that's also because I don't think I'll see the chat if you chat at me. So please do unmute yourself.
I can let you know if there's any questions.
Oh yeah, and let me know if anything comes up. Okay. Pretty much everything that I'll talk about today is based on the work of Alex Williams. Alex is at NYU now. He actually gave a talk here about a month ago, and does really interesting work at the intersection between statistics and neural data analysis. And he wrote this really very nice and clear paper about a very neat idea in 2018. The neat idea is what he called tensor components analysis. And this first figure from the paper is kind of going to guide our exploration today. I might go back and forth a little bit between this and the code, but for now I'm just going to talk through an example that we'll also kind of explore in the code later on. Imagine that you're doing some experiment and you're measuring from multiple neurons. I imagine that's the kind of stuff you folks do on day-to-day basis. Here there's spikes, but you can imagine that you've taken some fluorescence imaging and you've extracted out either spikes or waveforms, and you can kind of average across trials or smooth the data so that you get average firing rate as a function of time. So it's a continuous function of time from the spikes or from the fluorescence. And this is high dimensional because you're measuring multiple neurons, but it's also high dimensional because you're measuring in multiple points at time within every trial, and you're measuring in multiple trials. So in fact, in this data set, there are kind of multiple dimensions of the data that in each one, there are multiple observations, right? So there's multiple trials, multiple time points, and multiple neurons. And that really makes for a very rich data set, but also one that's really complicated to analyze. And so one of the methods that we'll start here with is principal components analysis. In principal components analysis, we take and we decompose the data into simpler, lower dimensional representations, and we add those lower dimensional representations that we call principal components to kind of re-represent the data. We take the data and we transform it in such a way that each component is much simpler than the original data, but you can put it back together to get the original data. You'll see what that's like. So something that people will do with this kind of data, because it has these three different components, and principal components analysis, and generally a lot of linear algebra depends on data being in a two-dimensional matrix form. This data is not a two-dimensional matrix, right? It's a matrix that has, or an array that has three dimensions: trials, time, and neurons. And so in order to use the standard linear algebra that uses two-dimensional matrices, you have to do something to the data. And the top here are two solutions that have existed for a long time. And we'll show these two solutions as a beginning of the code that we'll walk through. One is to trial average. That is to say, let's reduce the trial dimension. Let's consider each trial to be a copy of all the other trials. Let's assume that whatever we're doing in this experiment doesn't change over the course of the experiment, just for simplification. And let's average in every time point, gives us better signal to noise ratio, maybe. And so we get a matrix that is two-dimensional, has neurons on one side, on the rows, and time on the columns. That's a two-dimensional matrix, and that has numbers everywhere in it. And let's decompose that, and I'll explain what decompose means and what these blue and red things mean in just a second. Another option is to do what's called trial concatenated PCA. So we take that third direction of our data, the trial direction, and we concatenate along that direction. So then here in this case, we get something that's neurons by time. Here we get something that's neurons by time times trials. So concatenate together for every neuron all the trials. And there would be these weird artificial kind of like sewing points between, or kind of junctions between one trial and the next, where time is not continuous. But we can still do that and decompose that. Here, the advantage is that we haven't lost any of the information that was originally here through this averaging operation. So maybe if there's some change that happens over between trials, we can capture that too. But really, what we'll get to eventually through this tutorial is this idea that you can actually retain the three directions of the data and still do what I'll call a linear decomposition of the data, still boil down the data into these simple, relatively simple, low dimensional components, adding those components together into this three-way tensor. This is called a three-way tensor. So big, dense array of numbers is a tensor. And for the purpose of this conversation, at least, it's not super accurate. And a two-way tensor is a table like a matrix, like an image. One-way tensor might be a one-dimensional time series. Three-way tensor is a tensor that has three directions. It's a three-dimensional array that has neurons by time, by trials in this case. And what tensor components analysis allows us then to do is to decompose the data into factors that represent neurons, temporal factors within the trial, and then trial factors along the duration of the experiment. It allows us to say what are the variations in the data across combinations of neurons, time within the trial, and trials across the experiment. So we can pinpoint or rather decompose into the contributions of every neuron, the contributions of every temporal moment within the trial, and every trial to the sum total of this data. And if we're lucky, those contributions are not, we don't have to go really far in this series to have multiple contributions of neurons, of each neuron, multiple contributions of time, and multiple contributions of trial. If we're lucky and the data does have low dimensional structure, then it'll be enough to have a relatively small number of components that make up this three-way tensor. And we'll be able to decompose this into a relatively small number of components, and that will be enough to describe most of the variance in this data in a way that is highly interpretable. Okay, let me pause for a second here. What questions do people have so far? Okay, if no questions,
I have a
yeah.
kind of, I'm not sure if it's actually quick, but so kind of historically, some people amongst our labs have used this trial concatenated PCA. So we've, I think, like normalized like z-score to time series. And I like how you highlighted that the time series across trials are essentially stitched together. So I'm wondering if you could speak to what downsides you see to this method are. So I guess one thing is, could it be that where the time series are stitched together, if there is like some sort of consistent pattern where like the time series jumps from one value to another, that could be captured in the kind of the variance when you're running the PCA?
Yeah, I think there's not a huge downside to this, except that you still won't necessarily capture this third trial factor. You might capture these temporal factors, but you still won't necessarily capture these trial specific factors. And in fact, if the interaction is really complicated, it'll be hard for you to if there's really a complicated interaction between trial and time points, it might be really complicated for you to see this. I'll show an example of that in a second, where we can reconstruct temporal factors, but it's you can imagine a situation where I'll kind of describe how you go one level of complexity up, and now it's going to be very hard to pull that out.
I see.
So there could be additional complexity that's just not captured by this. So let me actually jump to the notebook. So this is the notebook that I'm going to work with here. I'll close this. I hope folks here are at least moderately familiar with Python code, but I'll explain as I go. Nothing here is, the complexity here is not in the software, it's in sort of the ideas. So let's create a data set like this. We'll have 20 neurons. The duration per trial will be 1 second at 1,000 hertz. I have 200 trials. And then for simplicity, samples per trial is duration per trial times the sampling rate. And I'm going to assign factors. I'm going to assign these 3 directions of factors. I'm going to actually start with the assumption that there are these 3 factors, and then I'm going to make this a little bit more complicated, and then I'm going to generate the data from the simulation. So here, I'm adding some noise in this, but here are my neuron factors. So I have 20 neurons, and here is kind of like the individual contributions of every neuron to the ultimately to the tensor. And you can see that there are 3 groups here, but maybe it'll be hard to tell apart some of them. There's some complexity here and some noise between them, even within the groups. I'm going to create 2 separate trial factors just to make things a little bit more complicated. But really these trial factors are not, and each one has noise. So I have 200 trials. One factor is, imagine like some component of the data that just keeps increasing with trials. And another component of the data that increases and then starts decreasing at some point during the trial. Imagine that depending on which of these components affects the particular data that you're looking at, you might get different kinds of effects here. Then I'm going to add sort of a temporal factor. Again, 2 separate, did I fail to run something? Oh, I failed to do this, sorry. 2 temporal factors. So this is within the trial. You can imagine, again, 2 kinds of effects that are happening where firing rate goes up and down in one case, and in the other case, it kind of goes down. And in order to create this kind of tensor of data, what I'm going to do is I'm going to take an outer product of these 3 factors. So I have a definition of an outer product here, but let's talk through this. So an inner product between 2 vectors is going to be important. An inner product between 2 vectors of the same length means that we take the multiplication of every element within the 2 vectors, and then we sum that up. That's an inner product, also known as a dot product. It's a very, or correlation. It's an operation that we do a lot in a lot of applications of signal processing. An outer product is slightly more unusual and is defined as following. Let's imagine that we have 2 vectors. One is n elements and the other is m elements. Then we can take the outer product between these 2, which will generate an n by m, so n rows by m columns matrix. And each element here will be so the first element is the product of this element, the first element in the first vector by the first element in the second vector. This one will be the first element in the first vector by the second element in the second vector, and so on. And then as we go down here, we're taking the second element in the first vector and multiplying it element by the other one. So we get within this, we get a table that contains the products of every element in the first vector with every element in the second vector. We get sort of a comprehensive, instead of, you can think of the dot product as taking 2 vectors and reducing that information down to one number. Here we're taking 2 vectors and we're expanding that to all possible products between the 2 elements. And that's called an outer product, in contrast to an inner product between them. So we can take outer products between, for example, here's an outer product of neuron by trial, neuron factor times trial factor 1 gives me one thing. And then to make things a little bit more complicated, so this, if I just ran this first row here, I would get something that is neuron by trial shape is 20 by 200, right? Because there are 20 neurons and 200 trials. So I get for every neuron trial combination, I get some, I can quickly show you the image of this thing because it's two-dimensional, something like this, right? So every row here is a neuron, every column here is a trial. We get something like that, but I like things to be a little bit more complicated. So I'm going to replace the second half of this by some other trial factor. Remember, I had two trial factors, these two trial factors. So I'm trying to make things a little bit more complicated. And so now the shape is still the same, but we get this slightly more complicated set of interactions where one thing is going up and then down, one thing is going up all the time. And then this is this thing. And now I'm going to take and do the second level of outer product here. I'm going to also introduce the time factor. So I'm doing the same thing here. I'm taking and I'm creating a three-way tensor, I'm taking my first two-way tensor, neuron by trial, and I'm creating a three-way tensor. And to make things more interesting, I'm going to have two separate time factors for two different populations of neurons. So here I'm showing you, because this is a three-way tensor and it's pretty big at this point, the shape is 20 by 200 by 1,000. So 20 neurons, 200 trials, 1,000 time points per trial. It's pretty big. Can't show you the whole thing, but instead I'm going to show you the first neuron, the 16th neuron. And you can see that I have trial, I have time. So this is the time. So this neuron behaves in a certain way over time and trials, and this one behaves in quite a different way over time and trial. So within this data, we have all these I've contrived a data set that has some complexity. Some neurons go all the way up, some neurons go up and then go down. Some neurons have this temporal characteristic, some other neurons have this temporal characteristic. And in our decomposition, we're trying to sort of build back to that these factors that I put in advance. So this is how I built up the data. And in fact, if we, our goal in some sense, when we do analysis of this data, is to discover from the data what is the role of every neuron. Some neurons might be responding more or less. We might have different trials. So we might have, maybe 100 trials through the experiment, some event happened and want to know that some neurons maybe respond to this event by changing the way that they respond. And then maybe there are temporal dynamics within each trial that we're trying to discover. So really what we're trying to discover is these things that I've contrived in advance by looking at the output of all of these outer products that I did. And I created these so that they have they're generated from outer products. So a method that is focused on decomposing the data into series of outer products should be able to rediscover this structure. That's our hope, at least. Okay, what questions do people have so far about the code, about any of these ideas?
So just to clarify and maybe get everyone on the same page, is it that you basically set some at the beginning, like some very general, like patterns, dynamics that that are happening across the population, across trials and everything, and then you're with those general like patterns, you're generating a synthetic data set of where you have like neurons, trials, and time that basically reflect those patterns.
Yeah, exactly. That's exactly right. That's what I did here. I started by putting in what I hope to eventually discover, the kinds of things that we discover when we do neural data analysis. I put those into the data, and I added some complexity because otherwise the data analysis won't be so interesting. And now I'm going to turn around and say, well, can we uncover these dynamics that I put in here, these distinctions between different populations of neurons that are doing different things and so on? Can I pull that out somehow by doing some decomposition of this tensor? Okay, what other questions do people have? Okay, so let me turn back to the slides, I think, at this point, and talk about a first important concept, which is the concept of a low-rank approximation of a matrix. So if we have a matrix of data, imagine that this is rows and this is maybe trial averaged firing rates. So each row here is a neuron, each column here is time point. One way to decompose this data is through what is called a low rank decomposition. The rank of a matrix is the number of rows or columns that are independent of each other. So here in this data, some of the columns have some dependence between each other. But if we want to approximate this by a low rank decomposition, we can create a matrix here. In fact, in this matrix, if you look at it, each column is a copy of the other columns multiplied by some number. So really, every column here is the same pattern repeated, but multiplied by some number. And sometimes that number is negative, right? So things that are positive in this first column might be negative in this sixth column here. But you know, these columns are just multiplication of each other by one number. And if you look down here, down the rows, you will see that also the rows are multiples of the same pattern. And so in this reconstruction, this low rank reconstruction, there is really just one column that is multiplied by other numbers. Each column here is a linear combination of just one column. There's just one pattern here. And each row is also a combination, a linear combination, or just a multiple by a number of all the other rows. There's just one pattern. And the way that we construct something like this is through the outer product. In fact, the outer product of two vectors is always going to be a low rank matrix. It's going to be a rank 1 matrix. If you take a vector of what we call loadings and a vector of what we call a component, then you get, just by the virtue of how this low rank, of how the outer product, how the math of the outer product works, you're going to get a low rank matrix. And in fact, every row here is going to be this component multiplied by some number here, the math of the outer product. And so one way to think about this is that maybe we find the best possible set of components and loadings so that this reconstruction is as similar as possible to the original data. And we'll see how we do that in just a second, like what's the mathematics of that. But for now, just think about the fact that we took data that was, you know, n by m, and we found n plus m numbers that represent as well as possible this data. So we've taken n by m numbers and turned them into n plus m numbers. And n plus m is, of course, much smaller than n by m. So all the information that is in this reconstruction, this n by m reconstruction, is already in these n by m two vectors here. And furthermore, the loadings you can think of as how much of this component there is in every neuron. So these loadings tell you how much each neuron correlates to this component, to this one component that we found to deconstruct the data. So we get the reconstruction by sort of matching, correlating every row in this matrix with this one component that we found that we think represents the trial average, roughly a trial average kind of variance in the data. Okay, let me stop here. What questions do people have about this idea of a low rank approximation matrix? So this is, I'm taking here one path. There are many ways to describe a principal components analysis, I'm taking one that relies on this idea of a low rank approximation, and we'll see how this ends up working in a principal components analysis. And this will also help us jump from the principal components analysis then to the tensor composition analysis. Okay, let me go back to the code then and try to do this. So, okay, so we talked about two strategies to make a three-way tensor into a two-dimensional matrix. The first is this trial averaged PCA. So here I'm creating trial average data. So I have 20 neurons, 1,000 time points. Here's one way to visualize that. Each line here is a single neuron, and each point here is sort of, or each trace here is kind of the trial averaged firing rate or fluorescence or whatever you want to think of this as. Firing rates of 2,000 is a little bit high maybe, so think of that as something that's measured over time. So we have a two-dimensional matrix now, and we can use something called the singular value decomposition in order to decompose this and try to reach these components that I described before. So our goal right now is to try to figure out how to get these vectors for a low rank approximation of the matrix. So here's a diagram of a singular value decomposition, of an M by N matrix. Sorry, I replaced my notation here, but M rows and N columns. It takes a singular value decomposition is a mathematical and numerical operation that takes and decomposes this matrix into three different matrices. A U matrix that is M by N, an S matrix that is N by N, and a V matrix that is N by M. And so it takes our data and it decomposes it into these three matrices that will be helpful in doing our decomposition into these low rank representations. So here I can do that. Here I've called them US and V. One thing to know about these matrices, something that's kind of useful about them, is that these U matrices and these V matrices are unitary matrices. That means that V, here, so let me show you the shape of these things. Shape, S dot shape, V dot shape is going to be slightly different than what I described before. U is 20 by 20. It's sort of on the smaller dimension of the data. It's a square on the smaller dimension of the data. This one actually has only 20 components. It's a vector, but we can turn it into a diagonal, into a square matrix of 20 by 20 in just a second. And V is 20 by 1,000. These are very closely related to the principal components analysis, but are not quite the principal components analysis. There's a relationship. I won't sort of go into the relationship between this and the principal components analysis, but I'll go to the principal components analysis next. This is starting with the singular value decomposition in order to show you that we can take the matrix and decompose it into these matrices, and that gives us information about the rank of the matrix, how many independent rows and columns are there in the data, and then it allows us all to compress down to the most representative low rank representation. So here are the singular values of this matrix, so-called singular values of this matrix. There's one very high singular value, and there's a pretty small singular value, and then all the other singular values are actually zero. So this matrix, first thing that we can know about this matrix is that because it has two non-zero singular values, that means that this matrix has rank 2. In fact, even after everything that I've done to try to kind of complexify the data is actually pretty simple. It's made out of you can think of it as having two independent rows or columns. So every row here is made out of two kinds of things, two separate columns, or vice versa if you want. Okay, and you can reconstruct this back. You can reconstruct the data back by multiplying u times here. This is just in order to turn this vector of singular values. I'm going to show you what that diag operation does. It creates a diagonal matrix. So it's zero everywhere except on the diagonal, and on the diagonal it has the singular value. So you see one large value, one small value, and all the rest are zeros. So when I multiply back u by v, I get back the original data. It's hard to see here, but in fact, this is the first neuron. This is the 11th neuron. I've picked out from the trial average data and then from the reconstruction of this two-dimensional data. And they're just completely overlapping because this entirely reconstructs the data. But I can do another thing here, which is to pick only the first singular value. So now I'm creating what I call a low rank. Let me split the cell here. I'm creating a low rank singular value matrix. So this is a matrix that has the same structure as my singular value matrix up here, but I've only retained the first singular value. I'm chopping off all the others. And this is where this connects back to my slides, in that now I'm taking the low rank of the data and multiplying back by u and by v, and I get low rank representation of the data. So now when I try to reconstruct the data, it's not as well reconstructed, right? Because it only has this one kind of overarching trend of the data. So it's trying to capture both of these kinds of things that can happen in time, just with one row and one column. So in effect, what I've done here is exactly try to find this component in those loadings. And if you look at this carefully, you'll see that what happened here is that I chose loadings as loadings the first, I believe the first column of u, and as a component the first row of v, because everything else here is zero. So it kind of nulls out everything else and just chooses from this and this two vectors to represent all of the data, and then multiplies them by the singular value to get back the reconstruction. So this is a way to get a low rank reconstruction of the data. You do an SVD, you choose few singular values, you erase all the other singular values, and you represent the data. Ultimately, under the hood, I've done this operation to get this approximate reconstruction now. And the reconstruction is not as good, but it still captures a lot of the original data. Okay, let me pause here. What questions do people have at this point? Let me show you this idea, but now using slightly different code. I'm going to use scikit-learn's PCA, the PCA principal components analysis.
Can I ask a quick question before you?
Yeah.
Is it possible to kind of give some context to what exactly U, S, and V are, I guess, representing? Like maybe trying to tie it back to the original kind of data dimensions, if possible.
Yeah, they're these things. They are a matrix of loadings and matrix of components. Let me try to plot. So this is U, and you can think of U as having, so it has 20 by 20, so it gives you the weights on individual neurons in, I believe, every every row here is a set of weights on neurons. You can see there are these 4 blocks that represent these 4 different groups of neurons. And then V is weights on time points, right? So when you put together, when you choose to do the first column of, the first column of U times the first row of B, maybe the transpose. That is not effective. Oh, because it's a dot product. I'm out at sea here. Maybe what I want to do is this. Yeah, okay, sorry, I'm out at sea a little bit. But the one way to think about this is that these represent each column here represents a column like this one, right? And if you go for the first 2 columns here, then you can represent your and that's kind of the next step in the slides also, is you can have several rank 1 matrices. So this this column times this row is your first summary, your first rank 1 matrix. This column times this row is your second rank 1 matrix, and this column times this row gives you your third rank 1 matrix. And when you sum them all together, you get a matrix that has rank 3, because you've summed together 3 rank 1 matrices, and those are by construction through the SVD, they're independent of each other. So this and this are constructed to be independent of this one, and this, they're all independent of each other. So finally, when you reconstruct, you get a more accurate reconstruction because we've included more of the data. For the SVD, because our data only had really 2 kinds of things, everything actually after these 2 columns, or after the first 2 rows of, this is not very, so these are the first 2 rows of, these are the first 2 rows of V. These are these temporal dynamics. One of them goes a little bit upside down from what we originally put in, but we have these 2 temporal dynamics that we put in, and V ends up representing these temporal dynamics. So you can think of that as like the column vectors. So what really the SVD gives us is very similar to this idea. There's like U's and V's, and we multiply them by each other. And if we put in, we have to put in the singular value so that the numbers multiply to the right amount. So just for scaling. And if we delete all the singular values except for one, we only get the first column of U and the first row of V, and we get this low rank approximation. And a PCA does something very, very similar. You basically take the original data and you ask, can I account for most of the variance in the data through a small number of rows and columns of row matrices and column matrices that each one has just rank 1. So I get rank 1 matrix, rank 1 matrix, rank 1 matrix. I add those together and I get a reconstruction. And the PCA will do this. In fact, the full PCA will have as many columns as you have in the original data, as many rows as you have in the original data, so that you get a completely accurate reconstruction. But sometimes you can get away with many fewer of these rank 1 matrices. You can end up with a reconstruction that has a much lower rank than the original data, and that's the dimensionality reduction part of this. You can represent, instead of representing the data as this very high dimensional thing, you can represent it as a set of much lower dimensionality things. let me show you how that actually plays out in doing PCA. So here's scikit-learn's PCA. Scikit-learn is a great Python library for machine learning. And the way that this works is that I'm going to tell it I want an object that explains 99% of the variance in the data. And I can fit that to the data, and then it tells me I can account for all of the data with one component. Because of this very skewed singular value structure, I can account for most of the data with just one component. So here is kind of the component, the temporal component that PCA tells me accounts for most of the data. It's kind of a compromise. If you think about it, it's a compromise between the two patterns that actually exist in the data. It's kind of average them together and says, you actually don't need more than that. So for this case, you end up with, I can also tell it, I want you to give me two components, and then it will give me two components that show me that. And in fact, I can put every, here what I've done is I've taken and I've transformed the data through this. I've basically done this operation of reconstructing through this thing, so that now I have for every neuron in my data set, I have the loadings on these two first principal components, and I can plot them, the first principal component, the second principal component, and you can see that every neuron falls along kind of like a straight line here. And it can show me that 99%, more than 99% is explained by just one dimension. And there's some amount of variance that remains to be explained by the second. I'll go quickly through this trial concatenate approach because I think we're almost, we have about 10 more minutes for this. So I'll just show you that you can do the same kind of thing, use the same PCA approach in order to deal with trial concatenated data. So here we're taking the other approach, this B approach, where we're concatenated the data together. And so we have trial concatenated data here. I've through the code, I've concatenated it. So we have 20 neurons and 20 or 200,000 time points, because I've concatenated 200 trials with 1,000 time points each. And I can fit the PCA to that as well. It takes a little bit longer, and I can get this kind of interesting pattern here. It sort of captures, very roughly captures these two, if you remember, I had two trial components, one that goes up and one that goes up and then down. It sort of captures these trial components here over a very long amount of time, but with lots of variation in each trial. So it kind of captures that. What I can do is I can say, well, maybe the first 1,000 points here represent the first trial, and then the second 1,000 points represent the second trial, and so on. So I'm going to take the reconstruction of the data into these PC components, and I'm going to reshape it back to the original data shape, and now I get these temporal components that I had. So while I'm doing PCA, I can actually reconstruct pretty well the temporal components within each trial. They're just kind of hidden inside of this larger scale node. So this is just to say that for some cases, the approach, this was your question, I think, before, for some cases, it's perfectly reasonable to do this time concatenated and then put it together in every trial in order to capture things that are true across every trial, like these temporal dynamics. You don't lose them. And then here as well, you can represent every neuron as a kind of like where it is in this PC space. And here, because we haven't averaged across trials, we get this the structure of the data. We can see that there actually are two dimensions to the data, and sort of these groups of neurons that fall into different quadrants here. Remember that I put in neuron loadings into 4 groups in the beginning. And so the trial average PCA kind of shows us this structure that does exist in the data in space and time, and neurons and time. Okay, but there's an alternative to this. and I'll just go to that alternative next, which is the TCA. So our data is tensors, and so what we can do is we can take the data as it is in its original three-way structure, and we can compose it into a low-rank decomposition of three-way tensors. These three-way tensors are going to each be composed out of outer products of three vectors. So if we take a three-way tensor and n by t or m by n by L tensor, we can decompose it into an m plus n plus L set of vectors. And when I do the outer product between them, I get the structure of this thing. That's the idea behind tensor decomposition, is to take a tensor and decompose it, just like we did with the PCA, into low rank matrices, into these low rank tensors. And the question then becomes, how many of these do we need? PCA takes and it can basically decompose the data into, it'll decompose the data fully. So you get a decomposition that has as many rows or columns as the original data. But here we're gonna, or the rank is as many rows or columns, the smaller of the two, as there are rows or columns in the original data. Here we need to decide in advance, and that's because the algebra completely changes when we go into this three-way stuff. There's no exact way of doing this. There's no equivalent of the SVD for three-way tensors. So now we need to use numerical methods in order to compute that. And fortunately, there's software that does this. The software I'm going to show you now is called Tensorly, and it does n-way decompositions of n-way tensors. So I'm going to use a method called parafac. Again, this is not an exact method. Instead, it uses approximations in order to find, in this case, I'm saying I'm going to give you the data, the three-way tensor, and I'd like you to decompose this into two rank one tensors, three-way tensors. So I get back from that. This takes a little while because it's doing some approximations and numerical operations. But at the end, I get three sets of factors that each have, you'll see this in just a minute. They're basically, under the hood, there are two three-way tensors that are represented by these factors that the TCA gives us. And this takes a little while because it's doing some math. I'll stop here. What questions do people have so far? All right, it's done. So now what we have is we have a 20 by 2 matrix, a 200 by 2 matrix, and a 1000 by 2 matrix. And if we take the first column here, the first column here, and the first column here, and we multiply and do an outer product, we get this first outer product. And then if we take, because we've asked for decomposition in a rank 2, we get another one. So if we take the second column here times the second column here times the second column here, we get the second rank 1 matrix. And the approximation, the TCA approximation of the original data is the sum of these two rank 1 tensors. But it's nice because also these rank 1 tensors each have some information in them. So if we look at this F1, what we get is the neuron components. In fact, what happened is the TCA divided the neuron components into two separate rank 1 matrices. If we look just at the first rank 1 matrix, it's basically saying there's nothing except for these two sets of neurons. It kind of eliminates the smaller sets of neuron weights that I put on these other ones. But if you add this, it kind of, in order to account for more variance, it kind of said, oh, actually, there's some stuff going on in these other neurons that have smaller neuron components on them. And same goes for F2, which gives you the trial by trial dynamics. You get the first component is this just ramp, and then the second one is, in this case, it's down and then up. But these kinds of these kinds of methods, one thing to know about them is the kind of sign invariance. You can think of this as if we multiplied this by a negative number, we'd be in this up and then down ramp. And then the third set is kind of the temporal dynamics within each trial. So what the tensor decomposition does is it kind of pulls apart the data into two low rank tensors, and then it tells you also each one of these low rank tensors is made out of, the first one out of weights on these first 10 neurons that have this ramp up operation and this kind of temporal dynamics. And that's exactly what I put into the data. You remember I divided the neurons into two groups. One group had a certain kind of ramping over time and temporal dynamics. And then the second set of neurons, these with smaller weights, have this kind of dynamics over trials and this kind of dynamics over time. And that's exactly what I put in to begin with. And then you can kind of look and see that the tensor decomposition also reconstructs the original data well. So this here, the top is the original data, the bottom is the reconstruction through the tensor decomposition. And if you look closely, you'll see that it's a slightly smoothed version of this thing, and that's because it removes some of the noise. So that's tensor decomposition.
With the sign invariance, is there any way to know the proper, if you need to multiply by -1 or not? Or do you have to just look at the data, the raw, I guess, population data, and like see what
yeah,
match?
it goes into you can think of this as going into the sign of this singular value, like does it kind of multiply to go up or to go down? Ultimately, when you do reconstructions, it fixes for that. And I think it's also stored inside of the PCA as some set of weights here. Yeah, in this case, I think the weights are 1 and 1, so I'm not exactly sure why this went negative. This and this went negative when they should have been positive.
You still get the rough shape, I guess.
Yeah, it gets you back the rough shape. It also get these upside down. Yeah, no, these it got here. It got the sign right. It's just here and here that it got the sign a little weird.
So it's 5 o'clock. Maybe it's probably some time for like a couple of questions. Unless is there anything? Is that basically it, Ariel?
That was basically I went through all the code, and yeah, there's not much more in the presentation except a link to this code, and I can share these. There are some links to blog post by Alex Williams and his papers, and also to this code if you want to play around with the code that I showed you. Play with that, and I'll share that with Charles.
Oh yeah, thanks for the nice breakdown of basically the Williams paper. Yeah, we have maybe if anyone has like a question or two. It can be related to this talk or like dimensionality or PCA in general.
But I should say, once this stuff kind of clarifies in mind, a lot of other methods, non-negative matrix factorization, some nonlinear dimensionality reduction methods, lots of things really rely on this sorry on this kind of. I really like this is not the way people usually explain PCA, but I actually think that this way is a really good way to think about PCA because you can build on it a lot of other things.
I guess I have like maybe like one general question is there, do you have any recommendations on like what you find is the best way to, if your goal is to reduce dimensionality with PCA? I know there's like the kind of an arbitrary cutoff of like taking 90% of the variance
Oh,
explained.
yeah.
Is there like a good way to figure that out?
Yeah, that was really tricky. What I would recommend to do is I showed how you reconstruct data, is to use a cross-validation approach to reconstruct data, and set that threshold. So this is about how many components do I need in order to, how many components are in this data? How would you know how many components there are in this data? And so one way is you could say, well, I want 90% of the variance explained and however many components I need for that. Another approach could be like, well, I want something that I can visualize on a two-dimensional plot. So I want two components, just to give me the top two components. Another approach is to look at these singular values and how they go down and just say, so long as there are kind of like, often what you'll have is you'll have like some high singular values and then a drop after something. And maybe you'll say, when I get this big drop, that's when I decide to cut off. Like,
So that's
it
like
doesn't
the,
matter, because for most real data, you'll get lots of singular values that are very small. So maybe you have some big singular values and then it drops, and then you say, okay, so long as the singular value. But the approach that I would take actually is I would say, take some of your data, do the dimensionality reduction on it, and then choose different cutoffs, and then reconstruct some other data that has different noise, and then see how many components you need in order to reconstruct this other data that has other noise. That's cross-validation. And that can be a really good approach to choosing number of components. Just go up until you're no longer reconstructing as well. So we've taken this approach in some kind of denoising approaches where you march up. So the more components you add, the more close you are going to the data. But if you're cross-validating, you're going to do well until you stop doing well, because you're starting to reconstruct noise, and then you'll start to go down. So our approach was just add more components until you reach a peak in this reconstruction, cross-validation, reconstruction function. Once it starts going down, just stop, because now you're reconstructing noise, and presumably everything past that is going to be noise. And that's very practical also for the tensor decomposition. You saw how long that took to do tensor decomposition. So you can't do a tensor decomposition to like thousands of dimensions. That will take very long. You just start marching 1, 2, 3, 4 until it starts not to cross validate. Yeah, one tricky bit there is that in contrast to PCA, when you do 1, then 2, then 3, then 4, you're not guaranteed that the first tensor component is always going to be the same. So in PCA, it doesn't matter if you do a PCA to like 1 component, 2 component, 4 components. The first component is always going to be the same, guaranteed, but not in tensor decomposition. You're not guaranteed that when you did 1 and when you did 2, the first component is always the same. So you need to be careful with that.
okay. If there's no questions, maybe Ariel, would you be free to like take questions like Slack or
Yeah, so I
email?
do, I'll put my email in here. And I do office hours for like data science questions. That's part of what the eScience Institute does. And I'm happy to talk to people about data analysis, things related to data they're working on, and questions. If this was a bit much for a Friday afternoon, I can understand. And if you want to go over any of this again over my office hours, I'm happy to chat with anyone.
Cool. Yeah. Thanks
And
again,
Charles
Ariel.
has my contact information. So if
Yes.
you didn't catch my email in the chat, ask Charles for my email.
Yeah. All right. Thanks again for joining us. I found the everything very enjoyable to hear about rehash. I've heard some of this already before. Um, but
Great.
yeah, hope everyone has a good weekend, and hope you do too, Ariel.
Yeah, happy Friday, everyone. Take care.
Thank you.